So we are going to continue on with our discussion of enzymes, but specifically now we're going to talk about what will influence enzymatic rate. And what do I mean by enzymatic rate? I mean the rate at which an enzyme can turn reactants into products. The rate at which an enzyme can do that can certainly be heavily influenced by different factors. You can see some of them listed here. We're going to go through these in just a moment. But I want to start off this by saying immediately that what we're doing here is we're trying to generalize. Because ultimately, to really understand this, you need to look at very specific enzymes and where they're located to understand exactly how these different things would influence that specific enzyme. For example, we're about to talk about temperature. There are certain enzymes where if you increase the temperature, that'll increase their enzymatic rate. There are certain enzymes where if you increase the temperature, that'll decrease their enzymatic rate. So please know, I'm going to give some examples, but we're not talking about specific enzymes in this lecture. We're trying to just generalize about what can affect the enzymatic rate. The first thing that we'll talk about that can influence enzymatic rate is temperature. The human body has an incredibly constant body temperature through homeostasis, through these negative feedback loops that we discussed previously. But if body temperature does rise significantly, or if body temperature does fall significantly, that can definitely have an influence over enzymatic rate. Now, there are certain enzymes where if it gets hotter, that will increase the rate of the enzymatic reaction, or if it gets hotter, that could reduce the rate of the enzymatic reaction. You really need to be specific about the type of enzyme that you're discussing. But just as a general rule, significant increases or decreases in body temperature is not good because it will change different enzymatic rates of specific enzymes, and that can have a very negative effect on the human body. It can actually kill somebody. But as I said earlier, luckily, our body does a phenomenal job, generally speaking, of regulating body temperature. So this is often not a problem, although it can be in extreme circumstances. The next thing that can certainly have an influence over enzymes is pH or acidity. This, like temperature, is going to be highly specific to the type of enzyme you're talking about and thus the location. So we have enzymes, for example, within the stomach. And the stomach is an incredibly acidic environment and it has a low pH. Low pH implies acidity, higher pH implies alkalinity or being more basic. So in the stomach, the pH is very low because it's very acidic. And we have enzymes within the stomach that operate and function at very low pHs. And if you were to make the pH either more basic or more acidic, it would both have a negative influence over the rate at which that enzyme could operate. Another way of saying that is decrease its enzymatic rate. The same thing is true for other enzymes, but they naturally operate at different pHs. For example, in the mouth where there's saliva, there are enzymes within the saliva that operate at pHs right around 7, which is pretty neutral. Now, again, if you were to increase the pH, make it more basic, or decrease the pH, make it more acidic, that would then have a negative influence over the enzymatic rate. In both cases, it would decrease it. The point is, having the right pH is very important for enzymes to be able to operate at the right enzymatic rate. And if you increase or decrease it, you can certainly have a negative influence over a specific enzyme's enzymatic rate. But you have to be very specific about the type of enzyme you're talking about and thus the location. So something else that can certainly have a big influence over enzymatic rate are cofactors. Cofactors are oftentimes vitamins and minerals, and not oftentimes, they really always are. They're vitamins and minerals. And what these vitamins and minerals can do is they can bind to specific enzymes. Okay? So here's a cofactor right here. Okay? What this cofactor can do is it can come over and it can bind to this specific enzyme. And what the cofactor will do to that specific enzyme is, we don't know for sure, but it probably ensures that the shape of that enzyme's active site is correct, or ensures that the active site can actually lock on to the specific reactant or substrate. If you look over here to the right, here is an enzyme. And the enzyme has that specific cofactor, which is a vitamin or mineral bound to it. So that would mean that the enzyme's active site is, let's say, orientated correctly or has the ability to actually lock on to its reactant or substrate. So all is good. The cofactor is going to allow this enzyme to be able to operate correctly. It's going to have a normal enzymatic rate. It's going to be able to turn its reactants into their products. But if you were to remove the cofactor, this vitamin or mineral, what would happen? Well, we can assume that without this cofactor bound to this enzyme, the active site would either become misshapen or the active site wouldn't be able to hold on to the reactant or substrate, and thus that would significantly decrease the enzyme's enzymatic rate. It wouldn't be able really to turn the reactant into the products. So cofactors can have an enormous influence over enzymatic rate. Now, let's talk about and look at an example. What you're looking at right here is what's called scurvy. Scurvy is caused due to a lack of the consumption of vitamin C. Vitamin C is in a lot of fruits and vegetables, and vitamin C is an incredibly important cofactor for enzymes that are involved in the synthesis of collagen, a very important protein for certainly skin-related tissues throughout the body. And much more than just skin as well. But that's usually what people see and what people think of when they think of scurvy. They think of, misformed and bleeding gums for the most part. There's a lot of collagen that exists within the gums of your mouth, which are really an extension of your skin. And if you don't consume vitamin C, then you don't have the cofactor that binds to specific enzymes that are involved in the synthesis of collagen within the gums. And this is what you end up with. So cofactors can be incredibly important to enzymatic rates. You can imagine that what would happen to the enzymatic rate of collagen-forming enzymes if there was a lack of vitamin C. Well, their enzymatic rate would significantly decrease. And why don't we say this as well? If you were to give that person vitamin C all of a sudden, what would happen? You'd be giving them the cofactor that they need. And that cofactor would then start binding to those collagen-forming enzymes. And of course, it would increase their enzymatic rate, and you'd be forming collagen again, and the scurvy would go away. So next in line are coenzymes. Coenzymes can have a big influence, a huge influence over enzymatic rate as well. Now, coenzymes are also cofactors. Okay? They're oftentimes vitamins or minerals that bind to an enzyme and allow that enzyme to function. So coenzymes are cofactors. But here's the difference. Coenzymes actually participate in the reaction itself. They don't just bind to the enzyme and allow the enzyme to function and perform its chemical reaction. Cofactors will actually participate in the reaction, oftentimes by removing some sort of chemical group. So two of the major cofactors that we will discuss in this class are what are called NAD and FAD. That's coming up very shortly here. And here's NAD and FAD. The main one that we'll talk about is NAD. And what NAD will do is it will bind to a specific enzyme within glycolysis, within the Krebs cycle, within beta oxidation, and it'll allow that enzyme to operate, but it'll actually participate in the reaction by removing hydrogens from the specific reactant. So this is another thing, like many things in these still somewhat introductory lectures, it's very hard to give context to these things because you're still kind of generalizing. But for the sake of an exam, you just want to know that coenzymes are cofactors, but the difference is they actually participate in the reaction. I think this will make much more sense to you when you actually see it in a real-world example, which, again, we are not far away from doing. That's actually part of this chapter. And since I've been saying the same thing with every other one of these, I should probably do it with coenzymes too. What is a coenzyme going to do to a specific enzyme? Of course, if that coenzyme is there, it's going to increase the enzymatic rate of that specific enzyme. And if the coenzyme is missing, that's going to decrease the enzymatic rate of that specific enzyme. Something else that can certainly have a huge influence over enzymatic rate is two things. One of them is how much enzyme is actually in the solution, and two, how much of the specific reactant or substrate is within the solution. So let's say that there's a particular amount, non-changing amount of an enzyme within the solution. Let's say that enzyme is amylase. That's an enzyme that breaks carbohydrates down. Okay, so there's an amylase enzyme that exists within the solution. And then what we gradually do is we increase the amount of carbohydrate that exists in the solution. Okay? So the carbohydrate is the reactant for that specific enzyme. So if you start increasing the amount of carbohydrate that exists inside of that solution, what is the enzyme going to do? Well, the enzyme is going to start breaking the reactant down into the substrates at a much faster rate. So that's the first part of this. You say, well, if you increase the amount of substrate or reactant within a solution, the enzymatic rate for a particular enzyme, whatever particular enzyme binds that substrate, right, is going to increase. So if we increase the amount of carbohydrate within a solution, the rate at which amylase is breaking that carbohydrate down is going to substantially increase. That's an incredibly important and basic principle for enzymes. If you increase the amount of the reactant or substrate for that enzyme in the solution, then the rate at which that enzyme breaks down that substrate or reactant will, of course, increase. I think that's fairly common sense. Now, what's not as much common sense is the next part, a little bit more challenging, but not that bad. So we're sticking with the fact that there's a finite amount of amylase within this solution. Eventually, you're going to get to a point where the enzyme, based upon the amount of enzyme in the solution, can't turn the reactants into the products at any faster of a rate. We would say that the enzyme is completely saturated. And you can see that's happening right here. At this concentration of carbohydrate within the solution, amylase can't break those carbohydrates down at any faster of a rate. The enzymatic rate is maxed out. We actually refer to this as the Vmax of an enzyme. The enzyme's completely saturated with substrate, and it can't turn the reactants into the products any faster. So the enzymatic rate is maxed out. Now, what our cells will oftentimes do, this isn't universal, but oftentimes what our cells will do to resolve this issue is they'll adapt. And they would like to be able to turn reactants into products at a faster rate at that given concentration. But since there's only so much enzyme within the solution, they're stuck there. They're at the enzyme's Vmax. So how do you resolve this problem? It's fairly easy. You just add more enzyme into the solution. the cells will actually adapt in this way. They'll actually increase the amount of enzyme within the cell itself. I don't want to give particular, in my mind I'm thinking, well, I'll give them some examples, but those examples are hard to give. I'll give them later on when we're going through real life examples. Let's just stick with generalizing here. So if this is amylase, and amylase is at its Vmax, so amylase is turning as much carbohydrate, is breaking as much carbohydrate down as possible. Right? The enzymatic rate is maxed out. How could we increase that enzymatic rate? Quite simple, add more amylase to the solution. If you add more amylase to the solution, now all of a sudden, the enzyme, there's going to be more enzyme in the solution, and you're going to be able to break more of the reactant down into the product. The Vmax will actually increase. Exactly why that is the case should be fairly obvious, right? At the Vmax, the enzyme is completely saturated with reactant, so it can't turn any more reactant into product at any faster of a rate. The enzymatic rate is maxed out. But if you put more of that enzyme into the solution, now there's going to be more enzyme that's capable of binding that reactant, and the rate at which the reactant can be turned into the products is of course going to increase strictly based upon the fact that you have more enzyme within the solution. Okay? I hope that makes sense. Another big thing is affinity. The affinity that a specific enzyme has for a reactant. This can be manipulated in a variety of ways, which we're actually about to discuss. But let's just say this. You have one specific enzyme, and you have multiples of that specific enzyme. Okay? So you actually have the same enzyme. Like you've got 1,000 copies of the same enzyme. A population of that enzyme has a much higher affinity for the reactants than the other one does. Why that is, we'll talk about in a moment. But it would be related, of course, to the active site. Okay, so here's population number 1 of the enzyme, and here is, let's say, population number 2. Okay, this is actually the same exact enzyme. Okay, but the active site is different. Okay, so population 1, population 2. You can clearly see that their active sites are slightly different. I'm not very good at drawing, but you can see those active sites aren't exactly the same. And there's 500 of each. Okay. The shape of the substrate actually looks like this. Okay. So which enzyme is the substrate going to fit into better? Of course, number 1 compared to number 2. I tried to draw it so that it looked like it was the shape of the active site of enzyme number 1 more so than number 2, if you're wondering. So what we would say about enzyme population number 1 is it has a much higher affinity for the enzyme, for the substrate or reactant than enzyme population number 2, right? Enzyme population number 2 has a lower affinity due to its active site for that specific reactant or substrate. So now let's take 500 of each one of these enzymes and put them into a solution. Okay. They're in the same solution, and then we put their specific reactant or substrate into the solution at a given concentration, which is right here. Okay. The enzyme population number 1, which we'll say is in green, the enzyme that has a higher affinity for that specific reactant or substrate is going to turn that reactant or substrate into its products at a much faster rate. Okay. The enzymatic rate of enzyme population number 1 is going to be much higher than the enzymatic rate of enzyme population number 2. And that is because the enzyme population number 1, right, with the more accurately shaped active site, can turn the reactants into the products at a much faster rate because they can bind the reactants so much better than enzyme population number 2 can. So the affinity that a specific enzyme has for a reactant or substrate will significantly influence its enzymatic rate. This is what we refer to as affinity. The affinity that a specific enzyme has for a reactant or substrate. If that affinity is higher, meaning it's more likely to bind it, probably due to the shape of its active site, then at a given concentration, it's of substrate or reactant, at a given concentration of substrate or reactant, that enzyme with a high affinity for that reactant or substrate is going to, of course, be able to turn that reactant or substrate into the products at a much faster rate. Due to its high affinity for the reactant or substrate, its enzymatic rate is going to be very high. And of course, on the opposite end, if you have an enzyme that has a low affinity ability to bind a reactant or substrate, at, for example, that exact same concentration of reactant or substrate, that enzyme with the low affinity is going to be turning much less reactant into product due to the fact that it's really kind of bad at being able to bind that reactant or substrate. It has a low affinity. So that whole conversation about affinity leads into this next part, which is about the regulation of enzymatic activity. Our cells have the ability to regulate the enzymatic rate of particular enzymes, and it's often done, not always, but oftentimes done through influencing their affinity for a specific reactant or substrate. And now let me explain and give you some examples. The first one that you're going to see a lot of in this course is what's called allosteric regulation. Okay? So here's an enzyme. This enzyme has an active site, which is right there, and it also has what's known as a regulatory site. That regulatory site is a location where another molecule can come along and actually bind to the enzyme. This isn't a cofactor or a coenzyme. This is a completely different molecule. And it comes along and it can actually bind to that enzyme on this regulatory site. And when that molecule binds to that regulatory site, it really typically does one of two things. It either increases the affinity that that enzyme has for a particular reactant or substrate, or it decreases the affinity that that enzyme has for a particular reactant or substrate. And how do you think it does it? Of course, by changing the shape of the active site. By changing the shape of the active site, the enzyme either can increase its affinity for a particular reactant or substrate or decrease its affinity for a particular reactant or substrate. Now, we have already talked about affinity, so what is going to happen in these different circumstances should hopefully make quite a bit of sense. So here, over here, this is the exact same enzyme. All three, red, blue, and green graph, they're all exactly, it's the same enzyme. It's either being allosterically activated, allosterically inhibited, or just normal. And in blue in the center, that's the normal activity of the enzyme. So as you increase the amount of substrate in the solution, right, the substrate or the reactant, the enzyme, of course, is going to increase its enzymatic rate. It's going to start breaking more reactant down into substrate. That's the way that it normally functions. Now, let's say that it's either allosterically activated or allosterically inhibited, and talk about what that means. With allosteric activation, what would have to happen is this molecule would come along, bind to its regulatory site, and result in the enzyme increasing its affinity for a particular reactant or substrate, right? It would change its active site so that it's more likely to bind a specific reactant or substrate. So if this enzyme is allosterically activated, let's look at a particular concentration of substrate, right here. This is the particular concentration of substrate or reactant. At this exact same concentration of reactant or substrate, look at the enzyme, right? The enzyme is breaking significantly more reactant down into product when it's been allosterically activated. And the reason for that, of course, is because right now it has a much higher affinity for the reactant or the substrate. That was done through what's called allosteric activation. Again, my brain immediately starts going into wanting to give you specific examples of when this happens, but I'm not going to, because you're going to see so much of it in this course that the goal is right now just to make sense of the concept. Okay? So let's do allosteric inhibition. Okay? This is the exact same enzyme. Okay? We're not changing the enzyme, it's the same enzyme, but a molecule comes along and binds to this regulatory site again. But this time, this is a different molecule. This molecule, when it binds to the regulatory site, actually decreases the affinity that the enzyme has for a particular reactant or substrate. Okay. So it changes its active site, actually, the active site of the enzyme, so that it has a decreased affinity for a particular reactant or substrate. So what's going to happen to the enzymatic rate at a given substrate concentration? Let's do this concentration right here. Look at this. Let's compare it to the regular rate at which it breaks the reactants down into the products, and now when it's been allosterically inhibited. When it's been allosterically inhibited, look at it's breaking significantly less reactant down into products at a given concentration of the reactant or substrate. Our bodies do this all the time. A better way of saying this is our cells do this all the time. They'll actually allosterically inhibit particular enzymes intentionally for various reasons. And what that really means is a molecule comes along, binds to the enzyme's regulatory site, and then decreases the affinity that that enzyme has for a particular reactant or substrate. So at a given concentration of reactant or substrate, the enzyme's enzymatic rate is significantly decreased. The rate at which it's capable of breaking reactants down into products is significantly reduced when it is allosterically inhibited. This whole thing, yet again, is called allosteric regulation, but allosteric regulation can either be activation or inhibition. And you'll see, as I've said many times now, you'll see a lot of this throughout the semester. The next one is called covalent modification or covalent regulation. Both mean the same thing. I know modification isn't written up there, but oftentimes you'll see this as covalent modification or covalent regulation. This is very, very similar to allosteric regulation, but it's also different. What it ultimately accomplishes is very similar, but the actual process is different. What oftentimes happens is a molecule doesn't come along and bind to the enzyme. What actually happens is a chemical group actually gets added to the enzyme through a covalent bond. What usually happens is a phosphate group gets added to the enzyme. Some enzyme actually comes along and it adds a phosphate group called phosphorylation. It actually adds a phosphate group or phosphorylates the enzyme. You can actually see that over here. And when the enzyme actually gets a phosphate group added to it, right, the enzyme gets phosphorylated, it can do the exact same thing as last time. That addition of the phosphate group can either increase the affinity that the enzyme has for a reactant, or it can decrease the affinity that the enzyme has for a reactant, which will then either increase or decrease its enzymatic rate at a given concentration of reactant or substrate. It's exactly the same thing as last time, which is why I'm kind of going through it so fast. Covalent regulation and allosteric regulation are really the same thing. You can be covalently activated or covalently inhibited, just like you can be allosterically activated or allosterically inhibited. And by you, I mean the enzyme. The difference is, of course, that in allosteric regulation, it's actually a molecule coming along and binding to a specific regulatory site. That's allosteric regulation. With covalent regulation, it's actually a chemical group being added to and bound through usually a covalent bond to the enzyme. right? The enzyme oftentimes, as I said, gets phosphorylated. It actually has a phosphate group added to it. And that addition of a phosphate group changes its affinity. Yet again, it's pointless to start giving you all sorts of examples of when this happens in the body because there's no context to what I'm talking about. So when this comes up later on in the semester, and it will many times, my objective here with this lecture is just so you've seen this before and it makes a little bit more sense when we get there. And here is the last thing. Oftentimes, allosteric inhibition happens through what's called feedback inhibition. Okay, that sounds confusing, but it's actually not that bad. Let's look at a process. We don't actually have to go through the process to make sense of this, what's called the Krebs cycle. So this right here is the Krebs cycle, something that we're going to talk about in a good amount of detail very shortly. For now, what I can tell you about the Krebs cycle is it's a series of chemical reactions that occurs. And this series of chemical reactions that occurs is done for the purpose of ultimately creating fuel to make ATP in what's called the electron transport chain of the mitochondria. And the Krebs cycle happens in the mitochondria too. So what can we say? I mean, you need to make a kind of an overarching point here, and that is the purpose of running the Krebs cycle is ultimately to create ATP. Now, it doesn't do it, it does it in one location, but that's a whole different conversation. It doesn't really make ATP directly. It kind of, to some degree, makes ATP indirectly. It creates the fuel that's necessary for making ATP. So we can say the function of the Krebs cycle is for the creation of ATP ultimately. All right. If ATP levels are incredibly high inside of the cell, right? ATP levels are enormously high. Do you think we need to be running the Krebs cycle at fast rates? The answer to that would be, of course, no. Right? If your gas tank is entirely filled up with gasoline, should you be adding more gasoline to your gas tank? Of course not. It'll spill all over the floor. Similar idea, right? There's plenty of ATP inside of the cell. We don't need to be running the Krebs cycle at any meaningful rate if there's already a ton of ATP inside of the cell. So what ATP will do is it will actually come into the Krebs cycle, this series of reactions, and bind to a very important enzyme of the Krebs cycle called citrate synthase. And when it binds to this enzyme, what it does is it allosterically inhibits it. It binds to its regulatory site and decreases its affinity for its reactants. And it does that by changing its active site. So when ATP is bound to citrate synthase, it's allosterically inhibited, and its enzymatic rate decreases. The rate at which it can turn its reactants into products decreases, and the whole rate of the Krebs cycle decreases, which is actually what you want here, right? Because there's plenty of ATP inside of the cell. You don't need the Krebs cycle to be making more ATP. Or let's be accurate, creating the fuel for making ATP. So when ATP levels are high, it actually reduces the rate at which the Krebs cycle runs. This is what feedback inhibition is. Feedback inhibition is you have a chemical reaction. It can be a series of reactions like the Krebs cycle. And what happens is the end product of that chemical reaction or series of reactions like the Krebs cycle, which is ultimately the creation of ATP, the end product of that reaction will actually come back into the series of reactions and slow down the rate at which it happens. Feedback inhibition happens all the time in basically all the cells of our body in different circumstances. All chemical reactions are done for the purpose of making something, right? For making products, ultimately. And what I'm telling you is that really the overarching purpose of the Krebs cycle is to sort of create the fuel for the creation of ATP. So we think of the true end product of the Krebs cycle, it's ATP. And if there's plenty of ATP inside of the cell, then we don't need this whole chemical reaction, this whole series of chemical reactions to be occurring. So ATP, the kind of end product that's created by the Krebs cycle, will actually go back into the Krebs cycle and shut down the rate at which it can run. That is the basics of what feedback inhibition is. And then you could think about it. When would the Krebs cycle speed back up? What would have to happen? Well, the cell would have to increase the rate at which it would be consuming ATP, which happens all the time for different cells. And if the rate at which the cell is consuming ATP increases, what will happen to the amount of ATP inside the cell? The amount of ATP inside of the cell will decrease. And then do you think citrate synthase will be allosterically inhibited anymore? No. Right? There'll be less allosteric inhibition of citrate synthase, and citrate synthase will then start turning more reactant into product, and the Krebs cycle will start running faster. And if the Krebs cycle runs faster, what will you get? I know I'm having trouble drawing arrows here. You'll get more ATP. If you run the Krebs cycle faster, you'll get more ATP. And then eventually, as the ATP increases to sufficient levels, it'll allosterically inhibit citrate synthase again and slow down the rate of the Krebs cycle. Okay? So that's the basics of feedback inhibition. Basically, always the end product of a series of chemical reactions will actually come back into the chemical reaction and slow down the rate at which it operates. So this is something I like to try to do at the end of most of my lectures, and that is kind of summarize and talk about the purpose of it, because I think it's incredibly easy in lectures to get completely lost in terms of, what you're actually talking about. What we've been talking about this entire time is what influences the rate at which enzymes function. Okay? Enzymatic rates. These are the main things that will influence enzymatic rates. Yet again, I tried to say this from the beginning, but I'll say it again. You can only generalize so much. The estimations of this vary significantly, but there's probably something like 75,000 different enzymes that exist inside of the human body. And how they'll be influenced by temperature, pH, cofactors and coenzymes. It's all incredibly specific to the type of enzyme that you're talking about and the location of that enzyme. But what we try to do is generalize and say, well, if you increase and decrease temperature, pH, change cofactors and coenzymes, that generally speaking can have effects over the enzymatic rates of enzymes. Okay, so as the class progresses, we'll talk about specific enzymes and specific circumstances, and we'll provide hopefully much more context about all of this.